Later on, she pursued her PhD at the Louis Pasteur University in the Strasbourg, France. Her PhD thesis identified an inducible mass in vascular smooth muscle cells. And after that, she migrated to Freiburg, Germany for a postdoctoral position. The last move was to Frankfurt in 1993, where she has held positions as postdoctoral fellow, assistant, as well as associate professor. She is now a full professor of physiology and director of her own institute, the Institute of Vascular Signaling, as well as the Center for Molecular Medicine at the Goethe University in Frankfurt, Germany. Currently, she is also a guest professor at Huazhong University of Science and Technology, Wuhan, China. Her research focuses on the elucidation of signal transduction mechanism in the vessel wall, in particular in the endothelium, and on the generation of several autocoids, such as nitric oxide and endothelium-derived hyperpolarization factor, which is called the EDHF. Her current projects aimed at elucidating changes in the metabolism associated with altered physiological states and pathophysiology, that is cardiovascular diseases and type 2 diabetes. This involves the use of mass spectrometry to identify metabolites and lipid mediators altered in different cells during the development of vascular diseases and wherever possible associated changes, that is the post-translational modification of the target proteins. She is a member of the Excellence Cluster Cardiopulmonary Institute Steering Committee and the principal investigator in the German Center for Cardiovascular Research. Further, Professor Fleming won several distinctions and awards, namely Green Shield Prize in Biochemistry, Dr. Paul and Silly Will Foundation for Early Career Scientists, Servier Research Award from the German Society for Microcirculation and Vascular Biology, Heinz Meise Award from the German Heart Foundation, Nitric Oxide Society Young Investigator Award, Arthur Weber Prize for the German Cardio Society Cardiac and Circulatory Research. Professor Fleming also won Sung Prize for the Medicine from Gießen University, and she is the president of European Vascular Biology Organization. She is also a member of both German and European Academy of Science. Professor Fleming also served on the editorial board of several prominent journals, such as American Journal of Physiology, Basic Research in Cardiology, Cardiovascular Research, and the European Journal of Physiology. Apart from that, she has also served as an associate editor in Journal of Vascular Research, Circulation Research. These are very big journals. She has also organized numerous meetings in vascular biology and medicine, apart from the organizing committee for the 100th centenary meeting of the German Physiological Society, which will take place in the Frankfurt this year. Professor Fleming also served in several scientific bodies and juries, namely German National Academy of Science Leopoldina, which is a very famous society, German Physiological Society, German Cardiac Society, German Society for Microcirculation and Vascular Biology, British Pharmacological Society, North American Vascular Biology Organization, American Society for Biochemistry and Molecular Biology, and American Heart Association. So this is the small introduction of my guide, and now I would like to invite my guide to address the gathering and share her immense knowledge in the field of cardiovascular physiology and vascular biology. Over to you, Professor Fleming.
Thank you, PRAVIR, or Professor Kumar. I hope I can live up to. I hope I can live up now to expectations. Can you see the screen properly?
No, it is not visible.
Okay. I have to finish out. I have to figure out how to do this.
Ma'am, to share your screen, there's an option below present now.
I've got it.
Yes.
I've got it, I think. Okay, is it now in presentation mode?
Yes.
Ma'am.
Okay, great. I was asked to give a couple of comments before starting about empowering women in science, and I know PRAVIR has already given you a short background, but I wanted to explain a little bit of the challenges that you can meet undertaking a career in vascular biology. And like he already said, I started off in the north of Ireland. which the rest of the UK, sorry, this is moving of its own accord, the rest of the UK sort of treats like a small province. Before moving to the mainland to study biochemistry and pharmacology. So I think some of the earliest things that I had to cope with were already at 18, sort of a huge sense of homesickness. A little bit of a change in language because we used to speak a dialect at home. And then I moved to France, a new language. I had to present my thesis in French. So in addition to the science, it was difficult to come up with communication with other people, so it was difficult to find a group of friends. Then I moved across the border to Germany, again another language, and that's one of the reasons why after moving in the early 90s to Frankfurt that I've really stayed there ever since. And like I said, I met a number of challenges like homesickness, the language I already mentioned. It's very difficult to start over again. It's almost like losing your personality. And I found that as a female, being constantly underestimated was a large problem. And it was at the beginning, some people met just previously, my group of technical assistants, but starting out, I find it quite difficult as a female to actually be accepted from the technical staff. It was difficult coping with some of the male colleagues who didn't believe that even in the 90s, a female should be interested in science. And I think not being put off when the tasks seemed too big was also a major challenge. And also learning to communicate with peers and juniors, like I said, with the language problem was an issue, but I think it's something that every female really has to learn to get around in science. And then I thought, wouldn't it be wonderful if science education was equally accessible to all? I think in India, you've gone a long way to that. And that the high percentage of females at the undergraduate and postgraduate levels were reflected in the percentage of female professors. And in Germany, we're a long way from that at the moment. Wouldn't it be wonderful if we all had more self-confidence and that we were all better at networking, that we all had more belief in what we're able to do, that we all had more courage to take bold steps, and that men did an equal amount of childcare and housework? Well, that's not going to work in any culture anyway. But I think if I have a message to anyone today, it's if you're aiming to reach goals, if you're looking for a career in science and you're females, don't be afraid of technology. I think you should put time in to have good grounding in the basics, but keep a pace with what's going on, with the developments that are going on, especially in the omics technologies and bioinformatics. And don't try to think like a man. I think females, we have a talent for out-of-the-box thinking, and I think it's really important to make use of it. Where possible, travel, see different labs, different countries. I've been to a lot of different places. Visited a lot of different labs, and I've gained a huge amount from that type of collaboration. And I think women usually are, if they can manage work and a family, they're usually good time managers. But you have to remember to balance work and family. And this comes in useful later on when you come to work with newer PhD students and postdocs. Of course, then you also have to train or teach your partner to be supportive. And I think I put this in red because I think it's the most important talent that anyone, be it male or female, can develop in a scientific career, and that's learning how to write. Because if you do the world's best experiments that are really worthy of a Nobel Prize and you can't write about it, you'll never actually be appreciated for what you can do. And I think this is the most important piece of information I can give to anyone. I also highly recommend, male, female, find a mentor. Find someone who's more senior than yourself, but who's willing to talk to you about issues at work or issues at home, and it will help you when it comes to making difficult decisions at the career or even at the personal level. And you have to learn and deal with constructive criticism. You have to be able to hear the good and the bad and turn it into something that's really constructive and useful for your career. And then I think my last piece of advice is simply that you don't just sit around, that you act on any of the advice given, and that you make a specific effort in developing your people skills in addition to your scientific skills to become, in turn, a mentor to someone coming on after you. Well, that's enough of the background. I think talking about science now is what I'd like to do, and I've got one boring title and one interesting title. The boring title is linking a relatively novel post-translational modification with vascular disease, and I'm going to talk about sulfhydration. And the better title I would call it is, wouldn't it be wonderful if? Now, wouldn't it be wonderful if you could take atherosclerosis like this is an example from the carotid artery in the mouse, and cure it, make it into something like this, decrease the rate of atherosclerosis. Wouldn't it be wonderful if you can take a mouse where there's no flow-induced vasodilatation and turn it into something that is close to a normal flow-mediated dilation response? And wouldn't it be even better, or almost similar to the wild type, wouldn't it be even better if you could do that in humans? If you could take people who have an endothelial dysfunction, cardiovascular disease, no flow-induced dilation at all, and give them something so that 4 hours later, the response is almost normal. And that's the pathway oh, why is this coming on again? Sorry, I don't know what's happening with that. So the pathway I'm talking about is a pathway of sulfhydration or transsulfuration. And this is a pathway whereby cystathionine is metabolized to cysteine by an enzyme called the cystathionine gamma lyase, or CSE. And this cysteine then can be metabolized by the same enzyme in the direction of pyruvate or in the direction of glutathione. And as a byproduct, this enzyme also generates hydrogen sulfide. And hydrogen sulfide is interesting because it's a gasotransmitter, it's a gas, it can diffuse throughout the cell. And it can also transcriptionally regulate or post-transcriptionally regulate or modify proteins. Now, this protein is interesting because it's down-regulated by laminar flow and up-regulated by disturbed flow. So it's linked, its expression changes with vascular disease. The H2S that it generates is vasoprotective.
Excuse me, ma'am. I'm really sorry to interrupt you, but I guess we are having a difficulty, like in yours. We are not able to see your slides. Yes, ma'am. Can you please present?
Now we can see your
Yes,
slide.
ma'am. Can you please put it in a full screen with the F5? You can in the slideshow move.
I'm having
F5.
Can you see it?
Yes, yes, yes.
In the correct mode, because to me it's wrong.
But it's a 12, slide number 12. Right now, the visualization of slide number 12.
Okay, I can't see it. So everything looks perfect. I can carry on because I can't see anything now.
Okay.
Ma'am, can you please present it in the slideshow part?
Yeah.
Okay.
So now it's okay?
Yeah. But Ingrid, this is slide number 12, which is visualizing us. It's a slide number 12.
I can't see the numbers. All I can see is it's the pathway.
Okay. Go ahead, please.
Okay. Stop me again if there's something strange.
Okay.
So this is a pathway that's regulated by flow. So it's regulated by changes in flow. The H2S that is generated is vasoprotective. And the thing you also have to know is that this enzyme can be phosphorylated. Okay? And why it's important is because this H2S and the H2N that it forms can post-translationally modify proteins. It can react with cysteine sulfenic acids, with cysteine disulfides, and with polysulfides on cysteine thiols. And when these changes take place in a protein, they can alter conformation, especially if it interacts with cysteine disulfides, to alter protein function. And the first example I'd like to talk about is an example from physiology, where we looked at what happens under normal physiological conditions with and without the activity of the enzyme. And I'm going to show you data that show you that under normal conditions, the generation of hydrogen sulfide by this enzyme is able to sulfhydrate proteins. One of them here is the HUR. It's an RNA binding protein that regulates the stability of some RNAs. When it's sulfhydrated, it cannot form an active dimer, so it remains inactive, so the RNAs are unstable. And in the presence of atherosclerosis or vascular inflammation, the enzyme is phosphorylated and turned off. This means that the active dimer can form, it can stabilize its target RNAs, one example being E-selectin, and that promotes atherosclerosis. Now, it's a complicated story, so that's the punchline, and I'm now going to show you the data behind that. So I talked about the enzyme being expressed in endothelial cells, and you can see here the red is the staining for the enzyme. It's all FAS in the aorta. And you can see that it's expressed highly in areas where there is a disturbed flow.
Sorry. We are not able to see your slides.
Sorry, I don't know what's going on.
Can you please switch on this presentation mode so that it may be because of this issue something is happening? Yes. Oh no, it's gone. It's gone.
Yes, I Can you see it now?
Yes, coming.
Yes,
yeah. Yes, CSE expression in endothelial cells. Yes.
Yes. Okay. Sorry. I don't know why it comes and goes because I'm not changing anything. The point is that when you look at areas of disturbed flow, you can see the expression is really quite high. And once you get to areas of laminar flow, you can see the expression of the enzyme is going down. It looks easier to see on a Western. If you do this in vitro and look at endothelial cells under static conditions, you can see a high expression or high expression of CSE. Expose these to fluid shear stress to mimic blood flow, you can see the enzyme expression goes down. And just as a control, the expression of the nitric oxide synthase will actually go up in these conditions. And if you work out the relative levels, you can see that the expression of eNOS and CSE, they're actually reciprocally regulated. And that's because both are regulated by KLF2. The eNOS is directly regulated by KLF2, and CSE regulation is indirect. It's KLF2 regulated expression of microRNA 27B. So, but what does the lack of CSE do to vascular biology? So as it was expressed in areas with disturbed flow, we looked at adhesion molecules. So we look at ICAM and VCAM and E-selectin, which are adhesion molecules or monocytes on the endothelial surface. And we found that even under basal conditions, the expression of E-selectin was higher in animals that lacked CSE, lacked the ability to produce hydrogen sulfide. And we saw this at the RNA level, at the protein level. You can also see it clearly in immunofluorescence in cultured cells. Interestingly, when you stimulate cells with interleukin-1 beta on top of that, you can induce the expression of ICAM and VCAM, but the expression of E-selectin in the knockout animals does not increase any further again. It remains at the same level it does in the absence of interleukin-1 beta, and that's also evident at the protein level. So even on its own, just the absence of hydrogen sulfide has a direct effect on the expression of E-selectin. Now we tried to figure out why that was. Does H2S directly sulfhydrate E-selectin? It didn't. We couldn't find any link at all that would help us understand why this enzyme was so intricately related to the expression of an adhesion molecule. So we decided to look at the interactome to see what was binding to CSE, and CSE is called CTH in this graph because it's the gene name. So we pulled it down and we looked at what was actually able to bind to it in endothelial cells. And one of the proteins of interest was HUR, human antigen R. And this is interesting because it's an RNA binding protein that stabilizes RNAs, and we knew that E-selectin is actually stabilized by HUR. And just this is the proteomic analysis, the same thing in Western blot. If you pull down the CSE from endothelial cells, you can see the HUR binding, protein binding to it. And if you look at the stability of E-selectin RNA under two conditions, these are on wild-type endothelial cells, and it's a chase experiment. So at this time point, we add actinomycin D to stop any translation. You can see that the stability of the RNA is decreasing, whereas in the endothelial cells that have a high HUR and no CSE have a much, the E-selectin RNA is stable over a much longer period. Now, is HUR sulfhydrated? And we can look at this by a number of means. The oldest type of assay we would use is a biotin switch assay, where we're able to label sulfhydrated cysteines, we do a switch assay, so we're able to demonstrate it with Western blotting. In the end, we can show that we do see sulfhydration of HUR in a wild-type mouse. If we have endothelials from a mouse that lacks this enzyme, the CSE, you can see that the levels are very low. DTT is added in this experiment just to show that this sulfhydration signal is specific, so it's our negative control. So you can also do the same sort of thing with immunofluorescence, taking an antibody against HUR and against sulfhydration. And this is a biotin switch assay. You can see the red dots show us that this HUR protein is normally sulfhydrated, and in the absence of CSE, the levels are really quite low. Now, why is sulfhydration so important? Well, sulfhydration, like I said, is able to affect disulfide bonds. So you can reorganize or reshuffle disulfide bonds and therefore protein conformation using H2S. And we found that cysteine 13 is the cysteine in this protein that is sulfhydrated. And if it's not sulfhydrated, something strange happens. If you mutate cysteine to alanine, we found that instead of forming an active dimer, this HUR was only able to form inactive monomers. So telling us that the sulfhydration of the protein determines whether it's a monomer or a dimer. And remember, the dimers are the active RNA stabilizing form. So we went back and looked at the wild type versus the endothelial cell specific knockout mouse. And we found that while in the wild type, there was a lot of monomer and some dimer, when we knock out the CSE, all of the protein was expressed as dimer only. So I think the next question of interest is, does any of this have a pathophysiological relevance? So the first thing we looked at was a mouse carotid artery ligation model of accelerated atherosclerosis. And this is an assay or an experiment that runs over 3 weeks, where we ligate one of the carotid arteries and look to see what happens. This is the non-ligated carotid artery, just as comparison. These are the ligated ones after 1, 2, and 3 weeks. If you look at the blow-ups, you can see that the expression of CSE is actually increasing in atherosclerosis. I said before, it's regulated by disturbed flow. And if we look at patients, if we take carotectomy samples from patients and look for CSE expression, we see the same thing. It's elevated in murine atherosclerosis. We see it's also elevated in human atherosclerosis. But the interesting thing is that if we look at the amount of H2S being generated by the enzyme, it's going down. It's going down really quite a lot, despite the increase in enzyme expression. In the human plaque, exactly the same story as in the mouse plaque. You can see the massive increase in the red signal in the plaque tissue, in the human vascular wall. And if we look at plasma, H2S N levels are going down. In the tissue, H2S levels are going down. So although there's more protein, it seems to be inactive. Now, why is it inactivated? I'm cutting this quite short to avoid showing you a lot of boring blots, but this enzyme can be phosphorylated on a serine residue that inactivates it. And the serine residue that becomes phosphorylated is serine 377. And this residue can be phosphorylated by protein kinase A, and that's, of course, activated by interleukin-1 beta. And you can see the effects on phosphorylation in the mouse carotid artery, where we see after 1 week, a weak phosphorylation of the enzyme, after 3 weeks, a really clear phosphorylation of the enzyme. And we can do this in humans as well, just using the biotin switch assay I showed you earlier. This time we're looking at the phosphoserine and CSE, and I think you can see the red dots coming up in the plaque of the human tissue and the endothelial cells. You can see that the enzyme really is being phosphorylated, and I've just added some red arrowheads here so you can actually see the difference in case your screen's too small. You can also do this at the level of the Western blot. It's the same method as I showed you before. Looking at sulfhydration, you can see that it's high in the non-plaque tissue and low in the plaque tissue. So it all seems to be consistent with inhibition of the enzyme. So in atherogenesis, we do have a phosphorylation of the CSE. It stops generating a lot of H2S. It means that there's no sulfhydration of HUR, so it can form its disulfide bond, become active, and then affect proteins like E-selectin. So physiologically, what does that look like? I said we performed the carotid artery ligation model, but this is what the atherosclerosis looked like after 3 weeks. If you look at the ligated arteries, you can see the huge plaque formed in the wild type or in the ApoE mouse. The ApoE mouse that has no CSE, the plaques were much quicker to develop and much larger. So the next question would be is, can we prevent that? Can we somehow address atherosclerosis in these animals in a rescue experiment? And we can actually do that because there are a number of persulfide donors that are available as therapy. And we use a compound called SG1002. And you can see from this massive accelerated atherosclerosis you see in animals without CSE, that we can basically decrease the level of atherosclerosis to actually below that seen in a normal ApoE knockout mouse. So what about humans? Can we take any information from this and take it back to the human situation? Well, I told you that CSE is generating H2S and cysteine from cystathionine. And if the enzyme is inhibited, we have the idea that cystathionine may then be elevated in the plasma, and that's exactly what happens. So what you're looking at is a graph of cystathionine versus H2S production in plaque-free, in plasma samples from patients that are plaque-free versus those that have atherosclerosis. In this blot, there are 75 patients. And you can see in black, the plaque-free group have a high H2S production, or high H2S levels in plasma, and low cystathionine levels in plasma. So the CSE is active, it's metabolizing cystathionine, producing H2S. In the plaque group, most of the samples are opposite. They're in a high cystathionine, low H2S group, so consistent with CSE activity being inhibited. And it worked quite well, as out of the 70 patient samples we had, only 5 were outliers and came up with the normal group. So one question is, where do you go from here? How can you take an observation linking one protein with cardiovascular disease? Well, we figured that H2S must be targeting more than one protein, more than this HUR protein. So we decided to scale it up. And this is what I meant about not being afraid of technology and making an effort to keep up with more modern approaches to look at older questions like post-translational modification. So we've scaled up the whole of the biotin switch assay. We combined it with something called PAIR-sulfide and dimidone to make sure we really can detect direct sulfhydration of specific proteins, specific cysteines on specific proteins. So we can really detect how many cysteines in a number of proteins in endothelial cells are sulfhydrated. And we work together with a bioinformatics group in the Max Planck Institute in Bad Nauheim to work out all of the data that we receive. And we decided to do this for a number of data sets. Data set 1, human endothelial cells cultured. They're the first passage, so short time in culture, static versus shear stress, to give us a CSE high, H2S high versus CSE low, H2S low group. And this was expected to identify targets of endogenously generated hydrogen sulfide. The second data set we looked at were endothelial cells exposed to shear stress to downregulate CSE expression. And then we treated them with the same donor that we gave to the mice. This is SG1002. And this data set was expected to identify targets of exogenously applied H2SN. And the third group we looked at were native human endothelial cells, isolated from plaque-free mesenteric arteries or plaque-containing carotid arteries. They were frozen within about 45 minutes of being removed from the patients. And this data set was to identify targets whose sulfhydration was altered by vascular disease. And you put all of that data together, we saw that comparing the cultured endothelial cells, static versus shear stress, with the plaque-free versus plaque endothelial cells, so the static versus the PF groups, they were quite similar. So the native endothelial cells and the cultured endothelial cells were actually quite a good model for each other. And in those cells, we saw most of sulfhydration was in the plasma membrane or in the cytosol, even in the nucleus, a lot of sulfhydrated proteins. In the SG treated group, however, we saw even more plasma membrane proteins, so that giving a donor targets proteins less in the cytosol but more in the plasma membrane. Actually, you'd really expect that if you're using a donor compound. If we look at all of the proteins that are sulfhydrated, we find over 5,000 cysteines in total, in over 1,500 proteins in all three data sets. Just looking at one data set of the native endothelial cells, we found over 1,500 cysteines in almost 600 proteins. So that's a lot of sulfhydration going on. And this is a post-translational modification that's been known for a while, but just how many proteins can be S-sulfhydrated was really not appreciated. Now, if we try to cut this down and we do some analyses about the proteins most affected, in all three data sets, beta integrins were the most highly sulfhydrated proteins. This is the number one position. I actually added HUR that I just talked about to the graph just to see where it comes up in this analysis. And these volcanoes show you in red and green the annotated beta 1 and beta 3 cysteines that were identified in each data set. And if you look at this in more detail, we find that almost every integrin can be S-sulfhydrated, but by far the most sulfhydrated cysteines come up in the beta 1 and the beta 3. So we decided to focus on that. And why this is interesting is because almost all of the cysteines that we picked up in these integrins are involved in disulfide bonds. And this was interesting, like I just showed you with HUR, the H2S is able to interfere with disulfide bonds and to reorganize them. And that's what seems to be happening with integrins when they're sulfhydrated. We found that the cysteines were in the IEGF3 and IEGF4 domains that are important for integrin bending, and in the hybrid domain that's important for ligand affinity. And the small heat map shows you the change, the gain and the loss of disulfide bonds that happens when we treat endothelial cells with an H2S donor. What does this mean? Well, it means that when H2S is there, that the integrin is an extended, open, ligand binding and active conformation. And in the absence of H2S, we have a rearrangement of disulfide bonds, which means that the integrin bends its legs and it's no longer able to bind to its agonist, like collagen, as efficiently. So that we should have a marked change in signaling. So we decided to visualize S-sulfhydration of endothelial beta-3 integrin. We'd already done it via the S-sulfhydrome. So we decided to use the same sort of imaging approach, labeling beta-3 integrin in red And the DAS2, this is a newer way to look at sulfhydrated proteins with a Western blot method, an immune histochemistry method. At the top, you can see endothelial cells under static conditions. They express CSE. You can see that there's a lot of yellow, so a lot of co-localization between the sulfhydration signal and the beta-3 integrin. Here below, shear stress. This is where there's little CSE protein, no H2S being generated. We see no DAS2 signal, no green signal, but still beta-3 integrin. So that we do see evidence of sulfhydration of beta-3 integrin in cultured cells. And on the other side, you can see the same thing in a plaque. This is human plaque material. And we can see that in the plaque-free artery, there's a lot of sulfhydration. And we do see a lot of yellow, which is sulfhydration of beta-3 integrin. Whereas in the plaque samples, we can see a lot of integrin, but very, very little evidence of sulfhydration. So what does integrin signaling do when you're thinking about flow-mediated responses? Well, one of the most impressive things in cultured cells is that they change their physiology or their orientation when you expose them to flow. So under static conditions, endothelial cells have a cobblestone morphology. They're not in any particular direction. You expose them to flow and they realign in the direction of flow. They become much thinner and much longer cells, and the Golgi reorientates and it localizes itself to just behind the nucleus. And you can use that to calculate a polarization angle, and you can see that endothelial cells will actually change and align with flow. Now, the IEC here means simply that the CSE as enzyme is not expressed. You can see that the cells maintain their cobblestone morphology. They do not change align in the direction of flow, but you can rescue that by overexpressing the CSE as an adenovirus. That's the green labeled cells. And you can see that all of a sudden now the cells that couldn't move here in the endothelial knockout of CSE now are able to align in the direction of flow. And something similar happens when you give an H2S donor. And you can map this in this type of spider web diagram and actually show the recovery of the realignment process. If you use, how is this linked to beta-3 integrin? We can show that if we put in a neutralizing antibody to beta-3 integrin, we can actually stop the realignment process of our endothelial cells. And it's actually maybe even easier to see in the blue and black diagram at the bottom that the cells really do stay in their cobblestone morphology if beta-3 integrin is not active. What about signaling? Beta-3 integrin is important because it's able to bind to G-alpha-13. And this is important because it leads to the inhibition of Rho-A, which in turn affects the phosphorylation of YAP, which then can move to the nucleus and suppress a number of genes. And we found that playing around with the CSE expression in endothelial cells, so using an endothelial cell specific knockout mouse, that we were no longer able to see that when we immunoprecipitate beta-3 integrin that G-alpha-13 no longer comes down with it. If we look at Rho activity, it's always elevated in cells that have no CSE. If we look at the phosphorylation of YAP, whereas it's phosphorylated in cells from wild-type mice, if there's no CSE in the endothelial cells, YAP is not phosphorylated. If it's not phosphorylated, it translocates to the nucleus to regulate gene expression. So by interfering with endothelial cell CSE expression, we've basically targeted all of the classical beta-3 integrin Rho-A YAP signaling pathway. So we have the situation where in physiology, where there's a lot of H2S there, that you have an extended form of the beta integrin leg, which is able then to bind to the extracellular matrix and to transduce signals that alter endothelial cells in response to the flow itself, like Rho-A. In pathophysiology, I showed you the changes that are linked to atherosclerosis before, where we have less CSE activity because it's phosphorylated. We now have a reorganization of sulfide bonds so that the integrin is inactive. which means it can no longer function to prevent the activation of RhoA. You can also link this to flow-induced responses. This is one of the examples I showed you in the beginning. It shows you flow-induced vasodilation in a wild-type mouse, so a normal response. You can see this type of dilation is completely gone if there's no endothelial cell CSE expression. However, you can take the knockout mouse and add the SG compound, which is an H2S donor, and you reach a response that's very, very close to that you see in the wild-type mouse. You can also do something similar. You can take a wild-type mouse and give it an anti-beta-3 antibody. You can see that flow is actually reduced. So it confirms the importance of this mechanism to flow-induced vasodilatation. And of course, it doesn't do anything in the CSE knockout mouse. It's something that's specific to flow, because if you look at agonist-induced relaxation, the response to acetylcholine, there's no effect at all of sulfhydration. So this really is a mechanotransduction process. And of course, if anything affects flow-induced dilation, you expect it to affect blood pressure. And these are experiments looking at blood pressure in mice. It's an inducible endothelial cell knockout. So we do that by giving the animals tamoxifen on 5 separate occasions. And then we wait to see that the protein levels decrease. And when that happens, we start to see within a week that the blood pressure of the animals that lack CSE starts to increase, and it stays up there as long as the animals then live. So we have CSE regulating beta-integrin phosphorylation, and because of that, it's affecting flow-induced vasodilation and in turn blood pressure. Now, can we take all of that, which was all done in mice, and take that and put it again in a human situation? Well, the first thing we had to do is we had to find a donor that we can actually give to humans. The SG compound we were using isn't available to give to humans in Europe yet. It's been used in the States. So we use a compound called methyl sulfamethane, or MSM. It's an H2S donor, or H2SN donor. And if we give it to a group of patients, you can see that we gave, with 12 patients in total, we gave 6 a placebo, and we gave 6 of them the compound MSM. And we found that within 4 hours of giving the patients with atherosclerosis, giving them the donor, their H2S levels were going up. Whereas the patients that received only the vehicle, their H2S levels stayed low. Then we looked at vasodilatation in the forearm, using a cuff to induce a flow, and looking then at to block flow, and then to look at reactive hyperemia, flow-induced dilation. And we found that in the patients that received the placebo, they stayed with no flow-induced dilation, no detectable response to flow. However, the same patients that we gave the MSM to and looked at the same time point, 4 hours after giving the compound, you can see that we've almost restored flow-induced vasodilatation back to control levels. And how can we prove that this is linked to sulfhydration? Well, we can't take endothelial cells from these patients, but we were able to take mononuclear cells and just look in general at the sulfhydration signal. And we found that the lack of sulfhydration that we saw in endothelial cells, we could reproduce in monocytes in the placebo group. But the group that received MSM, which restored flow-induced vasodilation, we also saw an increase in sulfhydration in the monocytes. So I've shown you that an enzyme in endothelial cells, the CSE, that generates hydrogen sulfide, it's crucial. It's crucial for mechanotransduction, and it's crucial for protection against atherogenesis. I started off showing you that the S-sulfhydration, or the attaching of the sulfur group to HUR, just as one example, was able to affect its conformation and to result in an increase in dimerization and activity, and that changing the dimerization of HUR can change its interaction with targets. I showed you data with E-selectin as an adhesion molecule, but there are a lot more, like cystathionine S, which is another protein that has been linked with atherosclerosis. And then I showed you how it's possible to scale up an approach like that and to look for what we call a sulfhydrome, which is the global pattern or the global overall view of how many proteins and cysteines can be sulfhydrated by this enzyme and how that can link to vascular function. And I think in total we had over 3,000 cysteine residues in almost 1,600 proteins. Just taking integrins as one example, because they were the biggest group of proteins we found, I showed you how S-sulfhydration is a critical determinant of conformation and activation. And then I also showed you that how this can be linked with altered flow-induced vasodilatation and mechanotransduction in cells, vessels, mice, as well as in humans. So I think my message here is to underline the importance of CSE and hydrogen sulfide generated in endothelial cells for overall vascular homeostasis and health. And this is probably the most important slide I have here. I don't know if Professor Kumar is going to recognize any of the people that are on the slide, but these are the people who did the work. They're a very, very good bunch of people, very talented group. Most of the work was performed by Sophia Iris-Bibli, who's just received an offer of a professorship here in Frankfurt, and a whole group of international collaborators from the bioinformatician Mario Lozo, but particularly Francisca Sigala in Athens, who did all of the human work. And with that, I'll finish and hope that there are some questions. So thank you very much.
Thanks a lot, Ingrid, for this great presentation, great presentation. And after 1.5 decade, I can see such a fully loaded presentation. Obviously, there are a lot of questions, and please don't call me Professor Kumar. I'm good for you. Please call me PRAVIR. So, Ingrid, there are a lot of questions, which is also in your chat box, but I can read for you. Like, Sudhanshu Sharma has asked a question. Ma'am, I have why S2S throat drops are not that much successful in therapeutics intervention of cardiovascular diseases?
At the moment, we're trying to find that out. There's a company in the US which has been making the compound we use in mice, this SG compound, and they have been performing a lot of clinical trials. So I can't really answer that at the moment. It has not been successful yet. We don't know why. We feel that maybe some of the donors may be targeting mostly plasma membrane proteins that we haven't seen. SG does target the beta-3 integrins, some of them don't. So I really can't answer that at the moment. We are working on different H2S donors to look at least in mice, how effective they are at replacing the endogenous H2SN.
Okay, Ingrid, I'll ask other questions from a student, but I request all faculty members, if you have questions, please ask to Ingrid, and then after that, I'll take care of questions of the students. Yes, Smita, Dr. Smita Rastogi Verma, please ask. Okay. If not, I can go for the second question from the Dia Advani. mRNA 27B are reported as oncogenic microRNA. Is CSE enzyme level links with cancer?
Yes, it has been. CSE is regulated in a lot of cancer cells, but you also have to remember that high levels of CSE are high levels of hydrogen sulfide can actually act as a mitochondrial poison. So that many of the enzymes in the TCA cycle can be sulfhydrated and inactivated, or their activity regulated. So we think that a lot of people have linked CSE expression with the Warburg effect.
Okay.
But in endothelial cells, we haven't seen much of an effect on metabolism in endothelial cells.
The next question from Meher Sahu. What are the dynamics and kinetics of sulfhydration in response to different physiological stimuli?
Good question. We see that it's quite quick when the enzyme is active. So we stimulate the enzyme, we see sulfhydration very, very quickly. So within a matter of seconds to 1 minute. We haven't looked at that many different situations. We've looked at flow, we've looked at stretch, we've looked at agonist responses, but agonist responses are not so important for sulfhydration. It seems to be mechanical stimuli, but they are quite quick. If the enzyme is active, the proteins are sulfhydrated.
Okay, Rashmi, you may ask. Rashmi, we want to ask question?
Very nice talk, Ingrid. I have no question for now. Thank you.
Okay.
It was a pleasure to hear you.
Ingrid, one more question from Meher. So, is there any mechanism or switch between sulfhydration and nitrosylation? It can work on same residue simultaneously or different pools of nitrosylated and sulfhydrated proteins.
We only did a preliminary experiment. I've been trying to find a student to do this. We tried once. We came up with some proteins that, yes, were nitrosated and sulfhydrated, but we never got any further than that. So we never tried to figure out if one is preferred under one condition and one under the other, because eNOS and CSE, I know there's a lot of confusion in the literature, are never normally expressed at high levels in the same cell. So when we go through the aorta, we find that CSE is high with disturbed flow, eNOS is low. If you go to laminar flow, eNOS is high, CSE is low. So we always find this reciprocal type of regulation. So we don't actually know at the moment, does nitrosation of one cysteine do the same thing as sulfhydration, or does it do something different? We really don't know yet.
Okay.
So we have few questions on the YouTube chat also.
Yes. I'm asking. So let her complete. Okay. So next question for you, Ingrid. That are you tired?
No. I'm fine. I'm good.
Okay. What are the relative contribution of H2S generating enzyme in various tissues other than endothelial tissues? This is from Vaibhav Oli.
It depends. Not many people have looked at organ-specific knockouts, so that you can extrapolate mostly only just from the global knockout mouse. We do know there's a lot in the heart, and that there's a lot of cardiomyocyte CSE. At the moment, we're trying to figure out what's in the heart, what's the endothelial cell component, what's the cardiac myocyte component. And there it seems to be certainly cardioprotective and have an effect on cardiac metabolism. We haven't looked at other organs, to be honest. We're a cardiovascular institute, so we tend to stick to the heart and the blood vessels. We haven't looked at anything else.
So another question from PhD student Sanchit Verma. He's asking, are sulfhydryl containing ACE inhibitor like jofenopril the future of medicine used to elevate heart diseases?
So I didn't quite understand that. So the question is, sulfur containing ACE inhibitors?
Yes. Are the future of medicine to reduce the heart diseases?
I don't know. Someone will have to try it.
Okay. And now this one student, he's a BTech student, she asked Simran Sharma why the CSC expression got increased in carotid artery in mice. And I'm taking one more question from Viral Srivastav, he's the BTech student. Ma'am, how does nitric oxide acts as a signaling molecule in vascular muscles?
Okay, two completely different questions. Why does CSE expression go up?
Yeah.
We think it's because of the disturbed flow. That it's going up, and certainly the more plaque tissue you have, the more disturbed flow, the more it goes up. The key thing is that the enzyme is just turned off so quickly. if any hint of vascular inflammation, any interleukin beta at all, it turns off. And that's why when this paper came out, it was 2019, so just after the CANTOR study, that showed that antibodies against interleukin-1 beta are really protective in vascular disease. And now we're really keen to figure out, is that linked at all to any protection against turning off CSE? The second question was nitric oxide in vascular, what was it, nitric oxide in vascular disease or in the vasculature? The nitric oxide is, everyone's learnt that it's all linked to cyclic GMP. I would say it's all S-nitrization. It's all transferring the NO to cysteine residues and altering protein function. But there's huge amounts of publications on how NO works in the vasculature.
Is there any other question from faculty?
No, I have a question.
Okay, so Ingrid, you please take some more sip so that you are ready to have some more questions.
Okay, go for it.
Professor Fleming, my question is more non-scientific. I like your caricature picture. It's really nice. So I just wanted to say who.
Okay.
So Dr. Simran, now you can understand why I'm so strict, because my training is like that.
No, I really like that caricature. It's really nice.
She is the strongest. Dr. Simran is the strongest woman in the vascular biology. Only 2 or 3% in the entire world which can compete Professor Fleming. Okay, so Dr.
I would disagree with that, but never mind.
Dr. Speaker, Dr. Yasha, please.
It was a very nice talk. I just wanted to ask one question, because you talked about inflammation and, ICAM. So ICAM expressions. So I was just wondering, in say the tumor microenvironment, where there is an increase in a lot of these inflammatory cytokines and increase in ICAM expressions and all. So how is it? I mean, what is your take on what is the differences in the vasculature that is also mediated by tumor-derived factors, because that is also a very important factor in changing the ICAM expressions in that milieu.
I can't say very much about the tumor environment, but we have been looking at how this pathway plays a role in angiogenesis. And at the moment, we've looked at physiological angiogenesis. We will start to look at tumor angiogenesis exactly for those reasons, but we haven't done it yet.
Okay, because there are conflicting reports on those aspects. So that's why I wanted to ask you about.
Yeah, you're quite right. You're quite right.
Okay, Dr. Yasha.
Yeah, that was one exemplary talk, I must say. Thank you very much, Ingrid. Now that this could be probably the last question before we end the session, would like to say a few words for encouraging the involvement or the contribution of women in science.
I think basically I said everything I could think of to encourage women, but I think the tips I had, I'd only like to repeat them again. I think one of the most important things is finding a mentor. Female, can even be male if you're pushed, but I think every young female scientist needs a shoulder to cry on, an ear to talk to, someone that's got their best interests at heart. And the other thing I would repeat is, don't be afraid of technology. Don't be afraid of leaving your comfort zone and doing something different. I mean, some of the data I presented today, I think my postdocs really had to teach me how to do it from the very beginning so that I actually understood it. But don't ignore technologies just because you're a female and you think, okay, it's too much for me. That would be my take-home message, I think.
Thank you very much.
Thanks a lot. Thanks a lot, Ingrid. It was mind-boggling, and I would really, I must say that after 1.5 decades, I have heard such presentation. So, and many thanks that you have accepted our invitation. And I'm really, I'm really touched after seeing you and your presentation. So
you're very welcome.
I will keep on calling you for such presentations in future also. So
okay.
I'm really, if anyone wants to have any remark, they are free to do for my guide, Professor Ingrid Fleming. If anybody want to say something about Ingrid, you please. Otherwise, we can switch over to the.
It was very nice talk, Ingrid, and I want to say you still look the same, the way you looked in 2004.
It's just this camera. It's such a big view.
Really, very nice to see you and hear you.
And you, Rashmi. And even Pravia.
So Ingrid, if you allow me, we can move to the next presentation. So will you be online or you will leave?
I will stay online for a few minutes and then I'll leave at some point. I have another meeting.
Okay. So over to Neha.
Hello, ma'am.
Oh, ma'am.
Hello, Daman, ma'am.
Hello.
Hello,
Neha.
So,
I'm good.
Daman, ma'am, I just want to welcome you on behalf of Department of Biotechnology and International Women Day. And the lady which just presented before was my PhD guide, Professor Ingrid Fleming. And she was also very close to Dr. Rasmi because we were. So over to you, Neha.
Thank you, sir. So thank you, ma'am. That was really an interesting lecture. So moving forward, I would like to repeat my announcements for the participants once again. The attendance is mandatory for each session, and link for the same will be available in the YouTube description and chat box during the lecture. There will be a feedback form towards the end of the last talk, which needs to be submitted for receiving the e-certificate. Please fill your names and email addresses carefully. We request you to post the queries, doubts of the lecture in the YouTube chat box. If the time permits, we will try to address most of your queries after the respective session is over. Thank you everyone for your cooperation. Now it's time for our last session of the event, and I request Professor KUMAR to please introduce our last speaker, Professor Daman Saluja, Director, Center of Biomedical Research. So please. Sir, please unmute yourself. Thank you, sir.
Good afternoon, everyone. So I'm taking this privilege to introduce another strong woman of our country, Professor Daman Saluja. She is the professor and director of Dr. Ambedkar Center for Biomedical Research, is one of the pioneer research institute in India. So Professor Saluja did MSc from University of Delhi, followed by MPhil and PhD again from the University of Delhi with the distinction. And her major contribution in characterization of SYN3B domains in especially interact with the other proteins like P53, the role of SYN3B, multiplex PCR-based diagnostics, because she is also involved in several developing the several diagnostics. Then characterization of HTF30, which has role in the mediated activated in the transcription, is a transcription factor. She has the expertise in developing several laboratories in India and multidisciplinary research. And 2006, he is involved in the as a faculty member in the ACBR, and since 2017, she was a director of this institute. This institute is growing very well right now. So I think she has many accomplishments. She has very good and very powerful papers in the peer-reviewed journals. She has around more than 110 publications in peer-reviewed journals. So over to you, ma'am.
Thank you, Professor Parveer, for such a nice session. And It is really nice to be at DTU for giving a talk. I had been associated with DTU for a long time. When it initiated the biotechnology department, I was in the committee, and we used to even go to conduct practical exams and sometimes even the lectures. And it has, see now DTU has come way with the wonderful faculty in Department of Biotechnology who are doing very good work, and off and on students have been coming from DTU for doing projects at ACBR. So we are interacting with them and finding that you people are all doing very well. Well, at AC, I did my, surprisingly, I would like to tell you, as Professor Fleming telling, we should not be afraid of changing domains and learning new things. I was trained as a chemist, and way back in 1986, I submitted my PhD thesis. And then, because I wanted to learn molecular biology, biotechnology, I decided to venture to USA. And at that time, plant biotechnology was in infancy. So I decided to switch to animal system. So I first went in microbiology and then went into gene regulation, which was my interest. I was working in gene regulation even in plants, with respect to plant hormones, but here to gene regulation, cell transcription, machinery, and learning a lot of techniques of molecular biology and biotechnology. And then my second challenge of coming back to India and joining this totally new center, which was just basically on paper, not even started. So the first couple of years was a struggle of setting up the department, not the lab, setting up the department, teaching is getting recognized, all the courses. So there was a lot of input by us, and some one or two later more faculty joined in. So together we started building up this center. And now I'm really proud. We have very talented faculty, and we have a lot of good facilities. We are doing a lot of good work. And many people have published in impact factor as high as 16, 17 from ACB. In their postdocs, many have publications even in impact factor of 30 or 35. But from ACBR, from our medical center, many of us have papers above 10 impact factor, which is a very difficult task coming from India to publish in such high impact journals. And a large number of papers above 5 impact factor. My lab, when I started working, since there was no infrastructure, so I decided to work on developing the nucleic acid based diagnostics for diseases that are more prominent in India. And many of the diseases, as you may know, are still identified by treatments based on symptoms and then given the treatment. And we now know that there are problems of, in many of these microbial infections, there is a problem of antimicrobial resistance due to many reasons. And one of the reasons is giving antibiotics, which may not really for that particular microbe, because the diagnostic was not run. It was based on symptoms, and many of the microbes give overlapping symptoms. So I started with that, developed a couple of diagnostics, even transferred the technology to an industry for commercializing, several patents and stuff. And we are also working on COVID diagnostics, interacting with some setups for point of care diagnostics. And even for leukemia testing, because these tests are mandatory for all patients to get done every 3 months. So we decided to develop an indigenous kit because the imported kits are very expensive and out of the reach of a large number of population. But my main interest understanding the gene regulation, which is an expensive research. So it took me a while to build up my lab facilities, build up department, so that I could pursue my first interest. And now I'm happy to, we have been doing some very good papers we have published. And but as I would as Professor said, that it is really great to have some mentor who can help, but unfortunately I didn't had mentors, but I had friends who off and on me and guided me. But having a mentor is really great, who can be always watching you and helping you over your in your in research career, sometimes only morally or sometimes even helping you in other ways. Yes, it is very important to find in India. We have another problem is women going out and making a scientific network, generating a scientific network, which is very difficult for Indian women to do, because we have a lot of family and social responsibility, and therefore it takes even longer for us as a women to really succeed in the research and scientific career. As you can see, there are a large number of women in life sciences. Many of them will go into the higher positions. We hardly have any vice chancellor, women vice chancellor, or secretary for DST, ICMR, or CSIR. Never, I've heard of, because they are not able to spend that much time because of their family responsibilities and many other reasons are there. And you really have to fight out and prove yourself that you are as good as men. So probably you have to work double the time or twice as hard as men or colleagues to prove that you have.
I'm just a moment, ma'am. Okay, she left already. She wanted to say hi to all of you. She left already. Okay, please carry on, ma'am. Sorry to disturb you.
Yeah, no, it's so. But still, I tell you, if you stay consistent, if you keep doing gradually your work sincerely, one day you succeed. It may be little later, but you do succeed. Hard work pays off, and I would say with these, please do not get discouraged with these hurdles. Some of the hurdles are same for males and females, getting funding and getting other things. But yes, we have extra responsibility. We have to work it out. We have to balance the two worlds, family and profession, and then work little hard to achieve something. So with these few words, I would like to start my talk. Can you allow me to share my screen?
Yes, madam, please. Yes, madam, please.
Okay. So I'm going to tell you something about our recent work on P73, which is, can you see my slides?
Yes, yes, ma'am.
Okay, all right. Okay, I will put it in the slide mode. So we started our work on understanding the role of P73. And then we found that it has an important role in invasion and metastasis. Can you tell me how much time do I have for the talk?
Madam, you have 40 minutes.
Okay, great. So I will take less than that. I will keep some time for.
Great, thank you, ma'am. Thank you, ma'am.
All right. So, so everybody is well aware of cancer. I think it is becoming alarming. Number of cancer patients in India are really increasing, but they are also actually increasing globally. It is not just. In 2018, 9.6 million deaths were reported. So you can say that there is almost 1 person out of 6 is dying of cancer. And many of the cancers are preventable, or at least can be diagnosed. If early, they can be managed, and the lifespan of the patient can be increased tremendously. Therefore, the diagnostics of cancer important, and for that we need to understand some of the basic mechanisms that are involved in the cancer progression. So usually we know that it is associated with cell cycle progression and defective apoptosis due to activation of proto-oncogenes or suppression of tumor suppressor genes. So in a normal cell, we have all these mechanisms which are kept in, kept alive. So if there is any error in DNA replication, cell repair it, or it will, if it cannot repair, the cell will undergo apoptosis. But in cancer, the cells fail to recognize those mistakes, and they keep on proliferating, in fact, adding more and more mutations as the cell progresses. So one of the common cancer worldwide is colorectal cancer. It is more prevalent. It was actually very prevalent in Western countries, but now increasing numbers are also coming up in India. So it has become a important concern. And metastatic colon carcinoma will result in kind of growing the tumor in the other neighboring tissues, urinary tract, in fact, in the urinary bladder, and in the other, sometimes going as far as kidneys and other organs. So we started with this work taking P53 as our model system, 3 protein, which is a chromatin remodeling protein, as our model system about 10 years back, but I'm not going to talk about that work at all. But while we were working, this is a standard P53 function. It is called guardian of genome. It's a very potent tumor suppressor protein. It is kept in check and the levels are kept low in the cell with the help of MDM2 protein. But when there is any DNA damage, or there is any abnormality or any kind of stress, including hypoxia, then this is released, and M2 is unable to degrade P53, and P53 is able to either results in cell cycle arrest, or if it cannot repair, then the cell will undergo apoptosis to remove or eliminate the cell. So there is all these phenomena is actually responsible for maintaining the cellular and genetic stability in the. So in the last 1 decade, I would say, people observed several P53 family members, and they found that these family members share a lot of functional and structural homology with P53. And these are P63 protein and 3 protein. All of them behave in a very similar manner. Used with any of the, will bring about very similar downstream activity. But then the question arises, what are their functions? Is there any specificity, or they are just redundant proteins? So we picked up P73 protein due to some of the reasons. P73 can bind to P53 response elements. Wherever P53 can bind, the binding site on the DNA is identical. But then a recent paper a couple of years back showed that there are independent targets of P73. When we look at in homology, it was highly homologous. So if I look into the homology in P53 and P63 and P73, you can see that transactivation domain, DNA binding domain, and oligomerization domain are highly identical. But there seems to be another domain in P63 and P73 which is missing in the P53. There is a at the C terminal end of the protein. Okay. So another thing was reported was that there are several isoforms of P73. There is a alpha form, there is a beta form, which lacks this C terminal domain, SAM domain, and a part of the other region. Then there is the N-terminal end of the protein is little different. This is delta N and alpha and delta N beta protein. Now the function of these proteins is just the reverse of the function of the normal length protein, because they lack transactivation domain. But in all other mechanisms, they are, all the three family members are very similar to ACT. And they even looks like they are inducing the similar pathways. But when we go into the details, then we find they are different. Like P53, P73 is also kept at very low levels, but it can get induced in response to the stress. And when we look at the tumorogenesis in detail, it looks like that role P53 is more in the beginning to generate tumor or to not generate tumor. If there is a mutant, then it is unable to suppress the generation of the tumor. But P73 roles looks like more into the later part of tumorogenesis. This seems to be suppressing the metastasis and invasion. And this will be clear from to you as we go along in our recent work. So although they bind to similar response elements, but there seems to be some kind of specificity for at least some of the proteins. Okay. So the question is, what is this difference and where is the specificity lies? Okay. Are there any unique targets of P73 that are giving it a functional edge in preventing metastasis and invasion, or is it just random? So another very interesting point that was observed when we looked into the literature was that although mutants of P53 are reported in most of the cancers, P73 is mutated rarely. It is only 0.5 to 1% of the cancers show P73 mutations. So this was also intriguing to us. So we decided to do a little bit omic study. So we looked into the genome-wide search of the genes that may be regulated by P73, some of the data that was available online, some binding studies, some chip assays that results that were available. So using a lot of published data and the gene ontology that available. So we, found some of the novel targets of P73. So there were three which there are actually here 4 that I have highlighted. Navigator 3, TIM3 and IGFB3 and FUR1. So I will talk about a little bit about these in my subsequent class. And interestingly, all these seems to be the proteins that are known to be involved in the invasion or metastasis. Okay. We found actually 7 long non-coding RNA which are also modulated, but we have worked on FUR1 in little more detail right now. The data for other 6 is kind of incomplete. So these are unique targets of P73 because if we delete P53, nothing happens to their expression. Okay? So they are not modulated. So for the student community, so basically what we did a lot of real-time PCRs and Western blotting in cell lines which were positive for P73 but negative for P53. Because we did not want a background of P53 response structure. So all the work we have done is done on the P53 null mutants. We use more than 1 or 2 cell lines so that to check whether these are actually kind of a universal phenomena or specific. And then we try to do knockdown of the target genes by using sRNA or by doing siRNA, depending upon the experiment that we need. So let me tell you a little bit about Navigator 3 protein. This is, the full name is neuronal Navigator 3, and it is predominantly expressed in nervous system. It is present on chromosome 12, Q21, and interestingly, it encodes a microtubule plus strand tracking protein. Okay, so therefore it belongs to a family which regulates cytoskeleton. And you remember that in cancer, the cells loses their shape. So cytoskeleton is very, very important. Okay. So Navigator 3 normally, and when there is induction on the growth factor, it kind of localizes to the plus ends of the microtubules and enhances the polarized growth and gives a characteristic shape to the cell, whatever is required. It is also very important in cell migration, Navigator 3. So if you deplete Navigator 3, the cells will show random cell migration, enhanced intracellular migration is this. So downregulation of Navigator 3 was also seen in the progression of many human tumors and was correlated with the prognosis. Means that the survival of the patient is predictable properly. That suggested that Navigator 3 has an anti-metastatic role. Similarly, TIM protein, it is a tissue inhibitor of metalloproteases. Many of you may have known that metalloproteases, MMPs, are actually induced during cellular invasion and migration during the later stage of metastasis, and they are released into the extracellular matrix. All several proteins degrade several proteins, making the cell conveniently move from one place to another. And there are natural inhibitors of these metalloproteases, which are also released by the cell, and TIM is one of them. So there is TIM 1, 2, 3, and 4. So we found that all are important, but this TIM3 binds to the extracellular matrix to the C-terminal domain of the inhibitor, and we are now, some of our recent experiments are suggesting that P73 has some role to play in the regulation of other TIM proteins also, but our results are not conclusive yet. So I'm not presenting here. So there are, so as I told you, it inhibits membrane matrix metalloproteases, and there are a large number of them. So it is definitely acting as a inhibitor for migration. So expression of TIC is important, and it also inhibits other proteins and angiogenesis by blocking VEGF, binding to the VEGF receptor 2. Now, angiogenesis is very important in tumor growth, and as the cells grow into tumor, inner tissues will not receive enough oxygen and carbon source, and therefore that hypoxia so generated will induce the VEGF and VEGF receptor formation, and therefore vascularization will occur. So TIM3 inhibits angiogenesis, so it will indirectly suppresses tumor cell growth. As I told you, in metalloproteosis, by inhibiting matrix metalloproteases, it inhibits the invasion and the cell growth. Okay. So what we did, we took these two proteins, and even for one protein, we are now we have a lot of data to support that. So we kind of a similar strategy for both these proteins. We looked at promoter region. As you can see, we looked into the 2 kb upstream. Transcription starts in almost 3 kb downstream. So remember that P50. So therefore, that in mind, we also looked for P73 binding sites. So the sequence is more or less similar, but as I told you that we are using P53 null cell line, so we were very sure that we will see the expression in response to P73 only if it is P73 driven. If it is P53 driven, we will not see the expression. Okay. So we identified first in silico using TF bind and Jasper program, and then we divided them into various blocks. So the reason for that was that we, so all transcription factor binding sites normally are half sites separated by few nucleotides. So we tried to look for that kind of consensus also, which was done almost manually. And we found that there was one cluster of P73 binding site, and one cluster was U2. So we upstream 1, upstream 2, and downstream 1 and downstream 2. So there was a gap in between. So we kind of took this region to clone, make one clone and another clone, and like that. And we gave them these numbers. So if these are, if these are really genuinely promoters for that, they should be able to upregulate or downregulate the gene to which they are present. Okay. So this is just to tell you what I meant was, there is a consensus sequence normally. This is the one half site, this is another half site separated by a spacer. And then P73 normally has a separate sequences, 10 base pair. And what we found was in U1 there were 6 such sites, U2 has 5, D1 has 5, and D2 had 4. So we took these regions and we started our experiments. So with this raw material that we had in hand, we started with a simple question first, that whether it is navigator P is regulated by P73 or it can be regulated both by P73 and P53. So we took P53 null cell line and we had very low expression. As I told you, in normal cell, the levels of P53 or P73 are very low. So there is hardly any expression of navigator 3 also. But the minute we give it a stress, by giving etoposide, which is a DNA damaging agent, So the navigator expression increases in null cell line, which were P73 cell lines. And if I overexpress P73, then there is more expression as this. Okay. And similarly, if I do P73 knockdown, then even if I give stress, there is hardly any change in the expression. But if I don't do knockdown, normal plus expression, then there is high expression. This is just like the same thing on present on a different scale. So this was by real-time PCR, and similar thing was shown by Western blotting. The expression of navigator 3 increases, beta-actin remains the same, but the P73 levels also increases the minute one gives etoposide. And if we overexpress P73 on a plasmid, then it also further increases. This is true both in the colon carcinoma cell line or in the another cell line we took, which was lung carcinoma cell line. So we see a similar effect within 24 hours, that there is a upregulation of both P73 and navigator 3 on giving the stress treatment. Okay, so this is the real-time PCR again, and this is the Western to prove the same. You can see here, if you have P70 knockdown, there is no increase in navigator 3, whatever was in the control 0 time, and so is true for P73 in both the cell lines, whether we take colon carcinoma or lung carcinoma cell line. So P53 effect. So that was also very important to prove that this navigator 3 is responding to P73, P53. Okay. If we take now P53 plus cell line, and we know P73, if it was also regulated by P53, then nothing should have happened. Knockdown of P73 should not have affected, because now there is in the background P53 is there. In our previous experiments, we didn't have P53, but in this experiment, we had P53. So in the presence of P53 and P73, you see the effect, which is expected. It could be by P53 or P73, but the minute you knock down P73, the expression of the navigator 3 goes down. You can see both in real time at the mRNA level and in the so this is not enough. This could be luciferase assay, we did chromatin immunoprecipitation assay, and we did site-directed mutagenesis. So this is the slide I showed you for navigator. We had U1, U2, D1, D2. So we took all these U1 and cloned it upstream of luciferase, U2, D1, D2, and we looked at the expression. And we found that it is U2 region which was more crucial. So the minute we give stress, if the luciferase activity increased only when we use this plasmid. In this, we didn't see much difference. You can see here, there's hardly any difference between plus minus stress. Okay? So that means similarly, when we look at the D1 and D2, we have, this is the expression of by the D2, which was upregulated, but by the D1. Okay, so this is within D1 and D2 and U1, between U2 and D2 also, we looked into the consensus binding site. So with the spacer, P70 binding site. So then we focused on the site-directed mutagenesis of these sequences to further prove that it is because of this. We also confirmed, as I told you, with the chip assay, that here you can see this is a positive control. So navigator gene promoter, when we do chip with P73, then and do PCR for the promoter, we see there is lot of P73 bound to the navigator 3. So I hope everybody understand what is the pattern immunoprecipitation. So what you do is you are trying to see whether P73 binds to a specific promoter. You kind of fragment the chromatin and do an IP immunoprecipitation antibody for the desired transcription factor. Here it was P73. So when we did P73 IP, we will get all the genome fragments to which P73 is bound. Okay? Now this bound is now deprotonized. We purify, deprotonize this, and now we have only DNA. But all these DNA had the P73 binding site. So we made the primers and amplified the P73 binding site. Okay. Now, if this had come due to P73 binding in the precipitate, then we will get a good amplification. The band will be strong. So as you can see here, in the as we go from 0 to 24 hours, the band will be increased. And here, so P73 chip say, and here is the P73 band here. Okay. So similarly, we carried out in the P73 knockdown. Here we did not see any binding because there was no P73. So we did not see, it is all background what we got. So it was done in both the cell lines. So it was a universal phenomena, not just one. Okay. And then when we did directed mutagenesis, we got in the U2, we got a, in the mutated one, we did not see upregulation even when we added etoposide. This is luciferase assay, but we also did with the, for both the cell lines, we have done that. U2 muted, different mutations, mutation only in one half side, mutation in second half side, mutation in both the half sides. Similarly, for the downstream promoter D2, mutation in one half side, second half side, and both. So you see that there is a decrease. So this proves that P73 specifically regulated navigator 3 gene. We carried out similar set of experiments for TIM3 protein by P73. So it is the same thing here. So again, we identified those upstream and downstream promoter sequences, P73 binding sites, carried out luciferase assay, and found that and carried out all the site-directed mutagenesis to prove our point that this is a specific binding. P73 binds specifically to the promoter region of the TIM gene also. And then chip assay, because it is done in vivo, natural operation, so it proves the fact that it was due to this. Okay, so oh sorry, this is okay. So there was an increased enrichment of P73 on the TIM3 promoter in a time-dependent manner, as we can see here. With increasing time, we found more and more assays in this, and similarly in the luciferase assay. So what we concluded from this part was that luciferase reporter assay and chip assay, that P73 binds specifically to the navigator 3 promoter as well as to the TIM promoter. Now then, what is the function of this? Okay. So we know that there is a, so in the beginning I told you, it looks like P73 has more important role in regulating the metastasis, which means migration, invasion. So we carried out some of these assays of migration and wound healing and cellular invasion in these. And we looked into the expression of metalloproteases, whether they are changed or not. So this is a classical, migration assay and invasion assay, probably some of you might have done. So you can see in the normal cell, it is very quickly getting filled. So you remove all the cells, scratch all the cells from here, and then you keep on going. And with time, within 24 hours itself, actually, you see most of it is. But when you have navigator, so in the control one, you have in 36 hours also, there is some small thing is left, which is not very dangerous. But when we have navigator 3 knockdown or P73 knockdown, this is in the 36 hours, it is almost gone. Okay. And if you have navigator 3 overexpression, but P73 knockdown, then also you see that it cannot be filled. So this suggested that navigator 3 is functioning downstream of the P73. Because if I do something which is downstream of P73, then it will bring about the same effect. So this is just a calculation how much migration had occurred in the different time points. Similarly, we did invasion assays in the wild type and the P73++ and navigator knockdown. You can see the cells all infiltrating and P73 knockdown. So you can see in both there was a lot of migration that occurred by the yes. So you can do the percent, you can plot the same thing by percentage or in the, how much is the closure. Okay. But then you remember that I told you that this is all invasion and migration requires matrix metalloproteases and many of the cell adhesion molecules. Okay. So we looked into the EMT markers, and as it was very clear, so as you knock down the navigator P or P73 knockdown, so this is just to show that we could actually reduce the levels of the two proteins. We found that P73 knockdown resulted in suppression of E-cadherin, both navigator 3 and P73, whereas N-cadherin increased. This is a very specific marker for tumor migration or metastasis. Vimentin also increased, fibronectin also increased, snail protein also increased. Okay. So all these shows are the markers for the cellular invasion, EMT transition, and they are well-established markers. Same thing we also observed with the matrix metalloproteases. If we knock down these genes, then we have upregulation of metalloproteases. Okay. So when P73 will be knocked down, you should remember that it will knock down, result in the like we will not have upregulation of navigator 3 or TIM3 or any other proteins that are specifically regulated by P73. So we see more dramatic difference in this case. So whether you do it as a time course study or just 1 point study, the results were the same, and we could see a very very characteristic modulation of matrix proteases, clearly indicating that P73 has a very important role in regulating the invasion and metastasis. Okay, so all these wound healing and migration assays and invasion assays clearly proved that P73 has an important role in suppressing the metastatic tissue. So if that is so, we should see a similar effect in the metastatic tissue. So we took some colon carcinoma biopsy samples and histopathology. And yes, indeed, we saw that the levels of navigator 3 and P73 in non-metastatic cells were higher, but they were low in the metastatic tissue. So P73 was low in metastatic tissue, but P73 was high in the non-metastatic. So you can see here, navigator is high in non-metastatic. 15 had shown high navigator 3 and high P73, but in the metastatic tissue, navigator was low in 10 of the samples out of the 15 that we used. And of course, navigator was also low, P73 was also low. And only 3 samples showed P73 little high and navigator, 2 samples showed navigator little higher than the cutoff value that we have done. So what does this tell? So what we found in a part of this study was that we identified novel downstream targets of P73. I have not talked much about TIM3 and long non-coding RNAs in this because of the paucity of time. And so P73 major role lies in regulating the migration and invasion, the upregulation of its target genes, navigator, and several non-coding RNA. And we know that it is really related in the cancer cells. Can we really use P73 at least to stop somehow the metastatic part? Because it is the three when metastasis begin, that's when the cancer becomes unmanageable. So in the stage 1 and 2, the cell the tumor is contained within that tissue. There are several ways of surgery, chemotherapy, and radiotherapy. Once invasive and metastatic, then can we think of something by which we can increase the levels of P73 or do something with the other downstream target and find out a way by which we can stop the invasion and this. I must acknowledge the work was done by my wonderful PhD students, Miss Apurva Uveja, who worked on mostly on Navigator 3, Neelam Bola, who is doing on TIM3, Chanchal, who is working on the long non-coding RNA, and interacted for the first 2 years as a DST Inspire faculty. And thank you for your attention. I will be happy questions from you.
Thanks a lot, ma'am, for the really power-packed presentation. And it's really made my day as well as my colleagues' day. I'm pretty sure that the presentation, after a long time, we have seen such presentations which are loaded with results. So means it's good that means Dr. Yasha has told me that, sir, we should organize some conference with the power-packed performance. So I'm pretty sure that both the sessions are completely power-packed performance. So, madam, there are some questions raised by students. So I'm saying the first question from the Smita Modi. She is asking, is P73 protein also linked and regulated by gut microbiota, and also it leads to the tumorigenesis?
First question, microbiota, we don't know. Microbiota itself is a highly complicated still, and there are a lot of work being carried out now in this area, but we don't know what is the cause and effect. So you have to remember that when we are talking many microbes, we don't know. So there will be good microbes, there will be bad microbes. And it is the homeostasis that is ultimately to lead to a particular disease because of the microbiota. So there are two back-to-back paper in Nature couple of years back, I think 3 years back, 4 years back, where they identified a microbe which would release a toxin, and they even purified that toxin and showed that if that toxin alone is given, it can induce cancer. Now the problem is that what that paper does not tell, whether that microbe thrived because the patient developed cancer? Was it the cause of the cancer? So where from that microbe came? Understand? So what made that microbe? That is not normally found in the microbiota, in the gut microbiota. So it must have come from somewhere. Was it like a environment pollutant or something, infection that they got, and it was otherwise not affecting the any, you can your digestive system or your respiratory system, but it just thrived there. So it is microbiota study is still in infancy, and lot more work need to be done to really understand how that is contributing to that. So cause and effect has to be really determined, which will not be that easy. It's like, doing another holistic research on the like you have on metabolomics, proteomics, post-translational modification, and so many. Cell is so complex.
Yeah.
So your second question was,
ma'am, that whether it leads to the tumorigenesis or not.
Yeah, P73 I told you is a family of P53 family member. So if there is a loss of P73, it is a tumor suppressor protein. So, but in the current scenario, whatever cancer tissues people have studied, they have found that in tumors, P53 is mutated or lost, but P73 is functional. So there was for a long time question was, why P73 or P63, although they have so much homology with P53, does not take up the role of the P53. So if they are, in many proteins, it's like that. If you delete one, the other will take over the function. But here it was not really functioning. And our results may give some clue that function of P73 may be later by suppressing the metastasis. So maybe there are mutations which are leading to the to this, which is resulting in the loss of expression of P73 in the later stages, not in the beginning of the tumor.
So before I take more questions from students, I request my fellow colleagues to ask questions to Madam.
It was a really nice talk. Ma'am, I have one question regarding autophagy and what is happening, because in the initial stages of tumorigenesis, it is known that the autophagy can cause the reversal of the EMT transition, and it can actually go back to the epithelial mode. And so, what is the role of P73 in that context?
So we have not looked into role of P73 in autophagy, but yes, we have in our mind. We have done some work on autophagy, not in relation to P73, but in relation to SINP protein, which we think also interacts with P73, the expression of genes. Because P73 or P53 has to bind to the promoter and downregulate the gene expression. So they normally recruit the repressor complexes. And because SIN3 is a very known, well-established chromatin co-repressor, which brings about, even HDAC proteins and all on the promoter. Therefore, we are quite hopeful. We are doing some experiments, and if I just share initial studies, we do see P73 getting interacted with the SIN3 and downregulating the gene that we are having. So in those aspects, the mechanism may be by gene repression, but whether once again, what we cannot right now say that whether it is directly functioning in autophagy or not, because we still have and looked at the genes which are involved in autophagy. So we will be carrying out this work, but right now I don't have evidence for that.
Thank you, ma'am.
A question from faculty. Okay, so I can go ahead with the different questions from the students, and one of the students, Dia Advani, is asking, the role of P53 is well known in the development of neurodegenerative disorders. So, is there any direct link of P73 for the Alzheimer's and Parkinson's disease?
No, as I said in the beginning, P73 really came into limelight only in the last 1 decade. And initially, people just thought that it is a redundant function. Like, it is known in many protein families. But the fact that P73 also regulates navigator 3 gene, which is involved in cellular shape, if we look into the nervous system and axons and all that, so we know that in the there are always philopodia and other podia being formed in the neuronal cells. It may have a function directly there, but I'm not sure that a direct function correlation has been shown for this mechanism, the formation of all these cellular extensions when they are done by the P73 regulated navigator protein. But yes, it is an interesting area of research which can be looked into.
Okay, madam. The another question from Sudhanshu Sarma, and he's asking, ma'am, is there any relevance of P73 with antibiotic resistance in Mycobacterium tuberculosis?
Your voice was breaking. Can you repeat the question?
Yes, ma'am. I'm just now it's audible.
Yeah.
Okay, ma'am. Sudhanshu Sarma is asking a question that is there any relevance of P73 with antibiotic resistance in Mycobacterium tuberculosis?
No, I have not come across any such correlation. Why would we have P73 have direct correlation with antimicrobial resistance? Because antimicrobial resistance by infectious agent is very, very different mechanism. So the microbe itself, develop certain mechanisms by, like efflux pumps are increased, drug degradation is increased, or they will have mutations in the protein to which the antibiotic binds. So it is more of a functional change in the proteins of the microbe itself rather than from the host cells.
Okay.
And in normal cell, otherwise also P73, P53 levels are very maintained in a low abundance. So I wonder if P73 should have any role in tuberculosis.
Okay. Ma'am, thanks a lot. Is there any other question? Thanks a lot, ma'am, for your wonderful lecture, and you are sparing time for us. So now, madam, I request you to please be here because there's some rituals like vote of thanks and all this thing. So over to you, Neha.
Thank you, sir. Ma'am, can I please request you to stop presenting? Yeah. Thank you, ma'am. So thank you, ma'am. Thank you, everyone. So with this, we conclude our session of the event, and would like to invite Dr. Yasha Hasija, Organizing Secretary and Convener of this one-day International Symposium, to present the vote of thanks. Ma'am, please.
Thank you, Neha. Surely we've come a long way, and today Nobel Prizes for scientific achievements have been awarded to dozens of women but still we have a long way to go. This symposium is a little contribution towards celebration of women in science and inspiring younger generation of women to take up career paths in STEM and scale up their contribution to the scientific community. On behalf of Department of Biotechnology, Delhi Technological University, I would like to express my sincere thanks to our Honorable Vice Chancellor, Sir, Professor Yogesh Singh. This symposium would not have been completed without his encouragement and bringing us opportunities to excel in our respective domains. I also wish to express my gratitude to our Head of Department and Chairman of the Symposium, Professor Praveer Kumar, for providing support throughout. I must also mention our deep sense of appreciation and a very heartfelt thanks to all the speakers, Professor Dr. Ingrid Fleming, Professor Daman Saluja, Dr. Vidhu Sharma, Dr. Gitanjali Yadav, and Dr. Shilpa Madan for joining us for the event from all over the world at varied time zones and delivering such inspiring sessions and guiding many women researchers in the field. I also thank all other faculty members of our department for their cooperation. I am thankful to the participants for joining us on this journey of encouraging and promoting women in science. Last, but definitely not least, I would like to thank my PhD students, Neha and Rajkumar. I must say, your work has not gone unnoticed, and we really appreciate your efforts towards organizing the symposium. I cannot thank everyone enough for their involvement and making this event a success. Thank you very much.
Thank you, ma'am.
Okay, thanks a lot.
Yes, sir.
You. And ma'am, I really appreciate your time. You have given us time. So once again,
you're most welcome.
Thanks to all of you, especially Dr. Yasha Hasija, because the entire event was her brainchild. She told me that okay, we should organize such a meet on International Women's Day. So kudos to you, Yasha. And at the same time, thanks all of my colleagues, wonderful colleagues and speakers. So now we can leave the session.
Thank you and good luck.
Thank you.
Thank you so much, sir. Thank you, ma'am. Thank you, faculties. Thank you, everyone. And with this, we end our event for today. And we request all the participants to please fill up the feedback form available in the YouTube chat description box by tomorrow 9 a.m. IST to receive your e-certificates. And thank you, guests and participants. Thank you, everyone, for taking part in our event. Hope you all had a great time.